It's a great pleasure to introduce our speaker tonight. It's Dr. Zineh from FDA. So Dr. Zineh has his doctor in pharmacology degree from Northeastern University. Then his residency at Duke University. And then he became a faculty member at the University of Florida. And from there, he arrived at the FDA, where he is for the last
1 year.
For the last 1 year.
Almost to the day.
So we're delighted to have him talking to us about personalized medicine.
Thank you very much for the invitation, and thank you to the troopers who are here on the Tuesday before Thanksgiving. So what I essentially wanted to do today was just give a high-level point of view on where I see the state of the science and practice of personalized medicine, or pharmacogenetics as a branch of personalized medicine, and maybe talk a bit about some of the barriers to translation and what we might consider doing to enable this technology or this way of treating patients to become a reality in the future. So by way of overview, I will talk about what I mean definitionally by personalized medicine, just some high-level concepts, if you will. I'll describe the environment, both the clinical environment and the regulatory and economic environment that's making us at FDA, as well as researchers and clinicians, pay more attention to this evolving area of precision or personalized medicine. As I mentioned, I'll talk briefly about the barriers to uptake and the critical factors for uptake, and then I'll end with just a few questions for us to consider. I don't have the answers to those. They're mostly to ruminate on. And of course, this is my standard disclaimer. These are my points of view and not necessarily reflective of official FDA policy. So let me start with a case that I think typifies some of the things that we're talking about when we're talking about making individualized treatment decisions for patients using technology like genetic information. So we'll start with a 33-year-old Caucasian woman. In April of 2008, she went in for a pelvic ultrasound due to about a year's worth of heavy and prolonged menstruation on the order of 7 to 10 days out of the month with associated weight loss. So she was diagnosed with a fibroid, no atypical Pap test. Oral contraceptives or birth control pills were seen as an option to help with the abnormal bleeding, but in the setting of intolerance to the bleeding. So it's a quality of life measure more than anything. On November of that year, she had a second pelvic ultrasound because of continued symptoms. There was some signal that the fibroid had possibly grown, but it was unclear if this was just a technical glitch in the technology, the way it was the image was taken. So the patient just said, why don't we just have this fibroid removed and solve the problem that way. So in February of 2009, a few months later, she had an annual test as part of her preoperative evaluation, and this time the cells came back atypical. And so D&C was done 2 weeks later, and she actually had a diagnosis of endometrial carcinoma. So this was interesting for a couple of reasons. One is she was incredibly young, so 33 puts her in the lowest 5th percentile of age of women that experience this. It's usually older postmenopausal women that experience endometrial cancer. Then it raises particular issues around screening for other diseases, genetic predisposition to other types of cancers, what to do about her child or siblings or parents in terms of screening issues. And then the other thing is the unique treatment dilemma, because the treatment for this is usually total hysterectomy and removal of both ovaries. And this is a woman of childbearing potential who wanted to still have kids. So a clinical decision had to be made with, let's call it imperfect data, but on a patient-specific level. And so she was diagnosed with stage 1A endometrial cancer, and the treatment for that, one of the treatments, at least a non-surgical treatment, temporarily, is treatment with megestrol acetate, or Megace. It's 80 milligrams given 4 times a day for a few months. The patient gets reassessed, and if she's responding, she gets continued on that for another 3 to 4 months. And so if the idea is that if the cancer is gone, the woman can try to have kids. If she doesn't have kids within a reasonable amount of time, she still has to get the hysterectomy. What I'm showing you here in this slide is the world's evidence behind this intervention. 27 articles, all case studies, representing 81 patients total, 28 of which were treated with Megace. This is the mean time to response, 12 weeks. This is the initial treatment response. About 76% of women respond initially. And then about 20 patients out of this 81 patients do get pregnant within a year or so. Now, the reason I'm putting this up is this is actually a real patient. This is my sister. This is her story. And the problem with this, as we saw it, was she was responding initially 4 months, but then she didn't respond at the end of 4 months. And so she had to have a hysterectomy and had to do that as her clinical treatment. The problem is that Megace, so not only was she non-responder, Megace is associated with the side effect of severe weight gain. She gained 70 pounds on the medication. So it would have been nice if there was some sort of genetic test or some information that we could have had prior to starting treatment that would have said, you're the 1 out of 4 patients that doesn't respond, and you're likely to experience side effects. That might have obviously changed the way we would have approached her therapy. And so this is just a classic paradigm. That's one example of an embodiment of what I'm talking about when I'm talking about personalized medicine, and that is, this is the common way we currently treat patients. You take a gross diagnosis, cancer, high blood pressure, high cholesterol, depression, you name it, and physicians will treat these patients with the same drugs that they're used to treating patients with, whatever they're comfortable with at the doses that they're comfortable with, when in reality there's a lot of heterogeneity in drug responses, as represented by the different colored people here. Responders are in green, non-responders are people likely to have side effects are in orange and blue, and the idea through is personalized medicine would take some genetic information, and before you start patients on medication, stratify them out into these 2 groups and approach treatment accordingly. And we know that that is a bit of an oversimplistic view because the technology has expanded to the extent now that we can not only think about genetic variations at the level of DNA, but we could think about changes in gene expression, changes in protein signatures, as well as patients' metabolites for the biological processes, all as potential tools to guide therapy. And so what this slide represents is a more streamlined, I think, a realistic manifestation of personalized medicine, and that is rather than taking a patient and giving them any of a number of drugs, A, B, or C, assessing whether or not they're likely to respond, which is almost always a no on the first go-around with monotherapy, and then adding another drug or switching to another drug or adjusting the dose and having this protracted cyclical way of treating patients through trial and error, we would use genetics to at least streamline this process. Maybe not necessarily get it right off the bat, but certainly eliminate some options for patients that wouldn't make sense. My sister's case being one example. So that's what I'm talking about when I'm referring to personalized medicine or pharmacogenomics. We're talking about making individualized treatment decisions based on genetic information to the extent possible, and using other non-genetic factors as well, of course. Now, a bit about the environment. I think we need to understand where we are in the present to identify where we might be going in the future. There is clearly no lack of interest in personalized medicine. What you're seeing here is references to personalized medicine in Google, in the Google world, since 2005 to 2009. And what you're seeing is exponential growth, maybe a dip earlier this year when the news of the economy was predominating essentially. But for the most part, steady. And if you look at the blogosphere, you're seeing a tremendous interest in personalized medicine. So clearly individuals are interested in this science and the potential it has for patients. And the idea is not only to respond to this interest from the FDA standpoint in terms of a regulatory and scientific agency, but to anticipate these emerging sciences and be a key player in bringing these through from a translational standpoint. There are tests and technologies out there that allow patients to make treatment decisions based on genetics. This is one of the FDA-approved tests. This is the amplichip that's made by Roche, and it tests for two drug metabolizing enzymes, 2D6 and 2C19. It tests for genetic variations in those two genes, which metabolize roughly around a quarter of all the drugs that we take. And so the idea would be that you can use a test like this and make your treatment decisions. And of course, there's been a huge expansion to beyond the clinic to more consumer-driven personalized medicine initiatives. These are the so-called direct-to-consumer programs and include things like Decode Genetics, which actually just went bankrupt, I think, this week, where for a couple $100, they would have told you what your predisposition is for certain diseases like prostate cancer and heart attack based on genetics. Genelex is a company that, amongst other things, has a $600 chip that tells you about your drug metabolizing enzyme signature. So this is absorption, distribution, metabolism, and excretion of drugs. 23andMe, for $400, was doing expansive genetic marker testing and told you something about your likelihood for experiencing diseases and your relationship between your genetics and several other traits, including ancestry. And then even for the bargain price of $350,000, you can have your whole 3 billion nucleotides that make up your DNA sequenced, and you can have that information at hand by a company called Genomics. So the point of that slide was really to show that the technology is there, it's in the public space, patients are starting to get used to it, consumers are getting familiar with it, and in some cases demanding it. So that's an enabling factor, I think. And of course, economics is really forcing us to think about the way we use medications. And this is an example that I like to use of a drug called Velcade, which is an anti-cancer drug used to treat multiple myeloma. So this drug, the interesting thing about this drug is that it only works in a very small percentage of the population, as do most cancer therapeutics. And the NICE, National Institute for Health and Clinical Excellence, is the oversight board within the UK that determines what goes on the UK formulary in terms of what they will pay for. And so this drug costs about $40,000 per patient per treatment cycle. And NICE said, we're not going to pay for this. We are not going to cover this drug. So the company, this is pretty innovative, in a risk-sharing model, they said, why don't you go ahead and pay for it, and then we will reimburse you, we will reimburse the government for the patients that the drug doesn't work in. So wouldn't it be nice to know, if you were a payer or a patient or a drug developer, who those patients are that are most likely to experience benefit from your drug? So this is an example of how the economics are really coming to play as well. This is a busy slide, so I won't focus too much on the details of it, but this slide, I think, illustrates the translational lag. And what I mean by translational lag is how long it takes to make your initial discovery to getting it in routine use in the clinic. And this is a survey of 32 medical interventions over time. And what you're looking at is seminal events in the history of these innovations, which range from the first description of the interventional class. They include the first description of the intervention itself, the first article about human use, first article on a specific human use, a highly cited article, and whether or not the intervention was partially or fully refuted. And the bottom line here is that when you look across, the median lag for making your initial discovery to the first high-level publication, enabling publication, if you will, is about 24 years. And so how do we expect personalized medicine to become a reality if we discovered the DNA molecule structure 50, 60 years ago? We just completed the draft of the human genome not too long ago. So there's an expectation that this is supposed to go into the clinic now, immediately, by clinicians, even by regulators and by third-party payers. But the reality is that it's going to take time. So how do we expedite this process? Why are we interested in pharmacogenetics from a drug development standpoint at FDA? I think this slide, the next two slides actually tell the story a bit. One is the so-called Valley of Death. This is the drug development landscape. And what you're seeing here is drug approvals at FDA over time in these blue bars. And what you're seeing is a clear reduction in drugs that are coming to the market. And it's not because the FDA is somehow imposing more stringent oversight. The oversight has always been stringent. What's happening is that the drugs are failing to meet their expected outcomes at several phases in the drug development process. What I have here is, to give you an idea, these are the phases of the drug development process, and they range from the healthy volunteer studies, which are the phase 1 studies, all the way to regulatory approval. And what you can see is that for the drugs that make it in humans, about 60% of them, only 60% of them make them to the next phase. Of those, only about a third, and of those, only about half. So overall, you're looking at about a 10% success rate for drugs to make it to, drugs that are tested in humans to actually make it and be approved by the agency. And the reasons are for lack of efficacy. So in early studies, we thought that it was going to be effective based on some signal that didn't translate out in the larger registration trials. Problems with differentiating your drug with against the comparator, or a safety signal that was problematic. Now, the interesting thing is we have examples from the pharmacogenetics literature on genetic determinants of all of those things. Here's a genome-wide association study of efficacy. Here's a candidate gene study of differential response. Here's a study on, here's a think piece on adverse events and how one might use genetics to elucidate the mechanism of adverse events. And of course, at the interface of all of this is informatics. So we have hope that this technology will somehow streamline that that development process and ensure a higher level of success. And there are some signals that this is happening. So in the cancer world, there are more and more molecularly targeted therapies, drugs that are developed because of a better understanding of the underlying etiology of disease. And in some cases, their use is restricted to certain subpopulations based on genetics of the tumor. And what you could see here is, this is the same figure as I showed, but this is for, the blue is the molecularly targeted therapies. And instead of a 90% failure rate, there's only a 50% failure rate. So 50% success is considered a home run here. So given the promise in not only drug development, but in clinical cases, as I've shown, what are some of the barriers to translation? Why aren't we seeing pharmacogenetics being more routinely taken up in clinical practice? And I have a couple of thoughts on that. Well, let me just say it's not because of a shortage of information. So this slide represents, in some way, the evolution of the mass amounts of knowledge that we're getting in the post-human genome era. Initially, we were interested in looking at genetic variations in single genes or a handful of genes that we thought might be involved in the either pathway of drug metabolism or the pathway of drug response. And we were looking at common variants that explained large effects. What we've gone to now is a more genome-wide approach where you can march along 1 million genetic variants in DNA and use informatics and statistical approaches to identify perhaps as of yet unthought about pathways for disease or drug response. And those are being done fairly routinely and fairly cheaply, actually. This is to put it in perspective, these are the number of these so-called GWASs, or genome-wide association studies. And what you're seeing here is the 22 chromosomes plus the sex chromosomes. And at each pinpoint, you're seeing a genome-wide association that was uncovered that achieved genome-wide significance at this level of p-value for the disease represented for that color. And the key is down here. I know you can't see this. But they range from everything from biomarker concentrations to AIDS progression to risk for lung cancer to response to the anticoagulant warfarin. So these data are out there. They're being generated in large amounts. And so a shortage of data is not the reason why this is not getting translated. And in fact, if we look at the number of publications and the number of genetic variations or SNPs that are involved in these associations over time, you see the revolution, the genomics revolution and the information revolution taking root. from 05 to 09, you can clearly see that the number, these are the number of publications per 6-month period that you're seeing. And you're also seeing the number of SNPs, or genetic variants, that have achieved significance, and the growth is exponential. And not only have we seen genome-wide association studies for complex traits and complex diseases, but we're also seeing them for drug response. And this is one example of a Manhattan plot, which, just to orient you to this slide, these are the chromosomes again, and each of these dots represents a p-value for one of a million genetic variations tested. And wherever you see a peak, you're seeing statistically significant genetic associations with the drug response of interest. This was flucloxacillin-induced liver injury. So this is a penicillin relative that has been associated with liver toxicity. And this marker region here is the HLA region that's important in human immunology. But we've had pharmacogenetic GWASs in many areas, and these are just some examples. Smoking's likelihood to have successful smoking cessation, likelihood to experience muscle side effects with the statin drugs, commonly used cholesterol-lowering medications, the likelihood for experiencing an antiplatelet response with the drug Plavix. So they're out there. So information is not the reason, but what is the reason for barriers to translation? And I think that they're all listed here, at least some of the ones that I think about routinely. One is the evidence conundrum. And what I mean by that is different stakeholders, whether they be FDA or clinicians or scientists or patients, may have different definitions about what is the evidence required to call something useful. The second thing is uncertainty regarding a clinical course of action. Let's say I report out a genetic test to you to help you make your decision, your treatment decision. Can you use that information? Is there a clear course of action? And is the genetics itself interpretable? Is there enough of a clinical genetic vocabulary out there that allows the routine practitioner to understand what to do at the patient level? I think another limitation is limited incorporation into clinical guidelines. And someone asked a question about level of evidence in evidence-based medicine, and we'll get to an example of that here and the issues around there. A fourth thing is what I call the abuse of clinical utility. And so it goes hand in hand with the evidence conundrum in that there is no uniform definition of what is clinical utility. There has to be some dimension of personal utility in knowing this information. And to the extent that those definitions are sometimes arbitrary, I guess at worst, and at best heterogeneous, there's never probably going to be a systematic demonstration of clinical utility for pharmacogenetics as a whole. Maybe in some one-off fashion for a drug gene test, that could be a possibility. There are other things that I think are less important as technology becomes more scaled. Assay validity, for example, time to results, and the cost. I think cost is essentially becoming zero for these tests when you think about the average cost of a drug for a year, say, or a lifetime. And of course, the complicating factor is the answers to these questions depend on whether or not you're a patient, whether you're a provider, and whether you're a payer. And so those perspectives will differ. And of course, another issue is education around these issues. So I think that's part of the story behind the barriers for translation. So let me talk about the evidence conundrum. The evidence conundrum simply states that we at some point want to take scientifically interesting biomarkers, whether they be genetic or other markers. We do this all the time. We've done it with blood pressure, cholesterol levels. At some point, they were interesting scientific markers that became clinically vetted somehow. The idea is that we would go from exploratory to valid for making clinical decisions. Now, the big debate is how do you go from the gray area of uncertainty to the black and white area of knowing what to do with that biomarker information. And that's where the evidence piece comes in. And so can you use genome-wide association studies of using retrospective data, prospectively collected, retrospectively analyzed? Can you use candidate gene approaches for randomized controlled trials that have been completed? A scientific question emerges after the fact, and you go back and test it and say something meaningful about a patient subgroup. And this is the KRAS story that we talked about a little bit earlier today. Or do you have to have a prospective, head-to-head, randomized trial of genotyping versus standard of care? Well, there's no simple answer. And the folks within the FDA, I think, can come down on either side of the question, depending on whether or not we're talking about safety, efficacy, or dosing. If we're talking about safety, you obviously cannot randomize a person to harm, and so we might be okay with looking at retrospective data. When it comes to efficacy, that might be a different question. And in fact, when we think about the evidence conundrum and what kind of evidence might be required, at least for regulatory decision-making, it actually might be quite different for clinical decision-making. What we've tried to do is map out conceptually the type of evidence that is required to make these different risk-benefit assessments. And just to orient you to this slide, what you're seeing on the x-axis is the impact of the event. So it ranges from insignificant to catastrophic, meaning if it's a life-threatening drug side effect, that's catastrophic. If it's some determinant of drug concentration that may not be important necessarily for the drug to work, that would be insignificant. What you're seeing on the y-axis is penetrance, the likelihood of experiencing the clinical endpoint if you have the genetic marker. And clearly, if you have a major catastrophic event for something that has a high, and your marker has a high penetrance, then this would be of major concern to us. And what you can see here, what you're actually seeing in the red circles is the proportion of patients that are likely to experience this adverse event or efficacy, diminished efficacy, or whatever the case may be. Just to give you one example, Tegretol, which is an anticonvulsant, has been associated with a severe life-threatening skin rash called Stevens-Johnson syndrome. It melts the skin right off, and it's irreversible, and people die from it. It's rare, but it happens. It's more prevalent in Southeast Asian populations, but it can happen in other populations as well. There was a case control analysis that came out of Taiwan showing that HLA genetic markers correlated with your risk for developing the Stevens-Johnson syndrome. It was less than 50 patients. And we relabeled the Tegretol label to include that genetic information with data from only 50 patients because that was so severe, and the likelihood for how else could you really study that except in this case control fashion. So that was actually sufficient evidence for FDA to update the label. With other things, you need larger prospective studies, and there are some examples here of the drugs that we've relabeled with pharmacogenetic information. Please stop me if there are any questions along the way. So as part of this relabeling effort, we like to think about what are the attributes and questions that a person needs to ask of a pharmacogenetic candidate to see whether or not there's a likelihood for uptake in the clinical community in terms of level of evidence, that is. These shifted down. So we described this in a framework called the pharmacogenetic pyramid, but the bottom line is, what is the clinical situation or need? Are patients not getting the drug because we're afraid of some side effect, that if we knew the genetic underpinnings of that side effect, it would make people more comfortable? Are we not pushing the doses high enough because we're worried about some toxicity that we can elucidate the genetic basis for? Is there a population that is not likely to respond to the medication because of genetics? And if we knew that, would it affect practice? That's what we mean by the clinical situation and need. The next thing is the marker association. How strong and plausible is this association? And some things that we think about are the statistics, magnitude and significance, whether or not it's been replicated in independent cohorts, whether or not there's a biological plausibility. Is the gene implicated in the drug's pathway somehow? Is there any functionality or mechanistic support? Is there some in vitro or in silico data that we can look at that would give us more confidence it's a real thing? Is there a gene dose effect, meaning do People that have 2 bad copies of the gene behave worse than people that have 1 bad copy of the gene, and do they behave worse than people that have 2 good copies of the gene? That would be supported. And then finally, support by analogy. So one that comes to mind is if you have a genetic association for a, I'm thinking of the Plavix example actually. I'm not sure if you're familiar with this, but Plavix is a drug that's used to treat 22 million people. And it's been the drug is inactive until it's activated to its active metabolite. And that activating enzyme is polymorphic. There's genetic variations in that enzyme. And therefore, poor metabolizers, genetically poor metabolizers, can't activate the drug sufficiently, and so they don't get as good of a drug response.
Why is the medication, why are they giving dosing with a prodrug and not the drug itself?
There was a lot. Yeah. So the question for the folks, in case they didn't hear, is why are they giving the prodrug as opposed to the drug itself? This drug is probably somewhere around a couple decades old, and the clinical pharmacology of the drug was not known at the time. Probably if it came in with the information that we know now, it would have looked a lot different. But it's a good question.
Okay, so it's not a formulation issue, just that's what we've been
Yeah,
doing. Okay.
yeah. Well, we now know that the active metabolite is highly, has a very short half-life. It doesn't stick around too long, but it sticks around long enough to knock out the platelets for the life of the platelet. But so those folks from genetic epidemiologic studies have been shown to not respond as well. The other thing of interest is that if you put people on a drug that inhibits that same metabolizing enzyme in addition to Plavix, so a 2C19 inhibitor, they also get a diminished response. And so that's what I mean by analogy. You have a drug interaction that mimics the genetics, and the genetics that mimics a drug interaction, and you see the same effect on clinical endpoints. That makes the story much more believable. And I'll show you a little bit about that, a little bit more about that in a second. So one of the other barriers to translation is that there are limited incorporation of pharmacogenetic recommendations into clinical practice guidelines. And to the extent that clinicians in the community use or are supposed to use these practice guidelines to make treatment decisions, evidence-based medicine, if you will, that is a real enabling thing if we can get pharmacogenetics into clinical guidelines. Now, this is a bit of a reality check on clinical guidelines here, because a lot of people say pharmacogenetics may not be evidence-based because there's not any randomized trials and all that kind of thing. Well, this was a publication that came out of a group at Duke where they looked at 22 or 24, something like that, clinical guidelines for therapeutic areas in cardiology, ranging from atrial fibrillation to heart attacks. And what they did was they classified the evidence behind their recommendations. Okay? And if anyone has worked with a cardiologist or has cardiology background, cardiology is considered the epitome of evidence-based medicine because everything is done by prospective, randomized, controlled mega trials of outcomes. So just look at the data, look at the survey of the data across these 22 clinical guidelines. Only 11% of the recommendations were level A, meaning that they were supported by multiple randomized trials or meta-analyses. 40% were level B, which is a single randomized trial or non-randomized study. And the clear majority were level C, expert opinion, case studies or standard of care. What that means is a group of folks got together and decided, this is the way I do it. I think it's the best way. Let's put it in the clinical guidelines. That's a bit eye-opening. Of the Class 1 recommendations, Class 1 recommendations are the ones that the professional society says, you must do this because it has been shown to be beneficial for patients. Okay? So if you don't do it, you're liable. Of the Class 1 recommendations, only 19% were supported by multiple randomized clinical trials. So the purpose of this slide is, I think, to demystify this idea that evidence-based medicine is always multiple prospective randomized clinical trials. That there needs to be some realistic assessment of pharmacogenetic associations and that they need to be somehow plugged into recommendations if they're robust enough to change practice, even if they're not tested prospectively against a standard of care. my own opinion, but I think it's a point that's important because it's a major criticism that I'm hearing on the streets as far as not having enough evidence to use pharmacogenetics in practice. The next barrier is the abuse of this term clinical utility, which we have a dirty word jar in my group, and if people use the word clinical utility, they have to put $1 in this jar because I think it's a bad word, and let me tell you why. I surveyed the PubMed literature up until a couple days ago for words that have articles that have clinical utility in the title or abstract. And what you can see is going back to the first mention that I could find to now, this just ramping up of interest in clinical utility. Now, of these just 609 papers from 2005 to now, that's just for this 4-year period, how many of those 609 papers do you think have the same definition of clinical utility? I didn't read them all, but I assume they're not many. And the point I'm trying to make here is that clinical utility is really thrown around as an escape hatch in some ways to not use innovations in medicine. And I think that could be dangerous because the guilty parties, researchers, clinicians, MDs and pharmacists, regulators and payers are all going to have different definitions of clinical utility and really need to consider what we mean by clinical utility from a genomic standpoint. And I would, again, make a pitch that personal utility, that is to say that some benefit at the patient level for knowing the information, even if it's not what we consider actionable in the traditional sense, probably should be incorporated. And that's the Secretary's Advisory Committee definition as well. These are a couple of evidence reports from the EGAP working group. So this is it's not the CDC, but it's a CDC-sponsored independent panel that weighs the evidence on pharmacogenomics. And so far, by my count, they've looked at several pharmacogenetic interventions, and for all of them, they have said that there's no demonstrated clinical utility. And I think part of it is around because of this ambiguity that we're talking about. So if people say the CDC doesn't recommend that's what the headlines read. CDC does not recommend pharmacogenetics. That could be problematic as well. We have plenty of examples of actionable pharmacogenetics, and this is just a summary slide of a lot of the actionable pharmacogenetics that we have out there. They're largely in the areas of oncology, and so these are the biomarkers of interest that you would test for, and based on these biomarkers, we make some decisions about which patients get which drugs. But I will say that actionable pharmacogenetics is not limited to the oncology space. We're starting to see more and more examples of actionable pharmacogenetics in cardiology and other therapeutic areas. We have a website at FDA that it's not comprehensive, but it's getting close, that characterizes all the drug labels that have some mention of pharmacogenetics in the label. Some of it is useful from an action standpoint, patient selection or dosing. Some of it is for information only. So those were the four, I think, big areas and big barriers for translation. And again, I don't think these bottom three are much of an issue. Well, so what are the critical factors for uptake? Can we learn from any of the pharmacogenetic successes to identify what are some key features, some commonalities that make a candidate more likely to be uptaken in practice? This used to be a prediction slide, but it's actually a reality slide now. So I used to read predicting the future. Pharmacogenetics will be used to sell health in one of three ways. Diminishing risk-benefit to improve safer use, to increase safer use in the market, establish genetically determined unmet medical need, and establish rational safety or efficacy dosing. And we add examples of all of these, and I'll show you just a couple. This is the abacavir story. I don't know if folks are familiar with the abacavir story. Abacavir is an HIV drug. It's a nucleoside reverse transcriptase inhibitor. And it's an effective drug. The problem with abacavir is about 7% of the population, Caucasian population, is at risk for a hypersensitivity reaction. Again, a skin rash. The problem is that if you stop the medication and then accidentally re-challenge the patient somehow, they get it again as part of a combination therapy or something, it could be fatal. So that's a problem when 7% of the population could die from a drug that you're using to save their life. What has been identified is that these HLA markers keep coming up over and over again. These are the ones that keep getting associated with adverse events. There's a genetic marker in this HLA gene region that predicts or correlates with your risk for that hypersensitivity reaction. And what you're seeing here is the test orders for HLA over time. And the association was published in 2002. The test was introduced in 2005. You start to see that by presenting it at the professional societies, including it in clinical guidelines, as we mentioned, high impact publication, that the test use started to ramp up. Clinicians started to feel more comfortable. And in fact, in markets that use this test a lot, like the UK and Australia, you can see that the market share for abacavir or abacavir-containing drugs is higher than for markets that don't really use the testing that much, for example, the US. So that means to me that clinicians are more comfortable to use the drug. We've modified risk-benefit relationship with this test. Now, how about this prediction that genetics will establish unmet medical need? So let's go back to the Plavix story, clopidogrel. As I mentioned, this drug is metabolized to its active metabolite in part through 2C19, which is genetically mediated. What you're seeing here is the pharmacokinetic, so the production of the active metabolite in people who are in carriers versus non-carriers of the reduced function genetic variant for CYP2C19. People who are poor metabolizers, for lack of a better word, are less likely to generate active metabolite, drug exposure. That translates to less of an effect on platelets. This is a platelet-inhibiting drug, so pharmacodynamics. And it translates into a higher event rate, okay? About a 50% higher event rate. I'm talking about cardiovascular events. So PK, PD, clinical outcomes.
How much of the reduced effect is just an imperfect correlation between the production metabolite and which allele of the allele you have, and how much of it is variation within? I guess I'm not saying that very clearly, but so is it that the reduced function allele is just not completely predicting? I guess so is that population is a mixture of some people who are responding as they would with the normal functional allele, and some who are responding very poorly, or is it all people who are responding?
Yes, I understand what you're saying.
Sorry.
So you're asking how deterministic is having this marker, pretty much, right? The answer is it's not perfectly correlated. In fact, we know that there are genetically poor metabolizers, if you will, that generate enough active metabolite. There are also extensive metabolizers that still have cardiovascular events, so it's by no means perfect. What I will tell you is that the benefit of the drug is rough relative to placebo, let's say, is about 30 to 40%. The excess risk is about that. So to me, it's not saying per se that people do worse on this drug. What it's saying is the drug probably doesn't work for them. Probabilistically, they still might get some benefit. What we don't know is that can you overcome the genetics by increasing the dose, right? And bypassing, because there's other ancillary ways to activate the drug to its active metabolite. But this, you may be interested on this side. There's a new drug called prasugrel or Effient. You may have seen some of the commercials. But this is, let's call it the cousin of clopidogrel. It works the same exact way to inhibit the platelets. The difference is a genetic difference. It's also a prodrug, but it doesn't rely on 2C19 heavily for activation. And so when you look, there's no difference in the genetic groups in terms of PK, PD, pharmacodynamic endpoints, or clinical endpoints. So this is, in a sense, the genetics have established an unmet medical need for clopidogrel, and understanding of the role of genetics in clinical pharmacology has led to the development of a drug that bypasses that situation. Of course, this is not an endorsement of this drug. This drug is associated with higher rates of bleeding and so forth, but it's just illustrative from a scientific standpoint. And then finally, an example of safety efficacy dosing. This is a, I'm actually going to skip this slide in the interest of time, but what I will say is this is actually through modeling and simulation and understanding of drug interactions that we've been able to make recommendations about this drug tetrabenazine, which is used to treat Huntington's disease, a very debilitating disease where there's not a whole lot of drugs out on the market. So if you go to the tetrabenazine label, you will see that based on genetics, we figured out a dose that you can go up to about 50 milligrams in most people before worrying about the genetics. But anything above that, you probably need to get a genetic test. It's metabolized by CYP2D6. So going back to the abacavir example, what are some of the critical factors for uptake? So what did we learn from abacavir and others? Well, I think this slide illustrates it. One, you probably need medical specialists who are more apt to be early adopters. So you need these people who are taking care of debilitating diseases with high mortality, the cancer docs, the HIV docs. Those are the ones that tend to be the most progressive in terms of personalizing medicine. We're starting to see more of it in cardiology, as I mentioned. Patients engaged in wanting to know more about their disease and treatments, patient activists, that's important. A networked community, usually by network I'm talking about information-wise. Incorporation into clinical guidelines, publication in high-impact journals. Regulatory agency recognition is more of an afterthought. We update the labels after all of this has been generated, so I think that's more, at this point, reactionary and not necessarily a prerequisite to having clinical uptake. And then, of course, prospective incorporation into drug development, like in the case of oncology, where you have a test and a drug developed at the same time for a specific subpopulation. That's clearly, that's terms for using the drug, so that has to have clinical uptake. Getting back to the evidence question, it's important to understand that reduction to practice requires practice. This is an example from Mass General Hospital in Boston where these folks are collecting DNA from tumors, and what they're using these DNAs for is to genotype these tumors or molecularly type these tumors and use drugs based on tumor typing rather than the site of the tumor. So for example, let's say I have a drug for colorectal cancer. And I have a patient, though, that has lung cancer, but their tumor looks like a colorectal tumor from a genetic standpoint. Can I use that drug that's approved for a different type of cancer to improve benefit, to achieve benefit in these patients? So these are very innovative approaches, and it's going to take some time. One thing that is becoming clear that I don't think anyone could have really predicted is that large pharmacy benefits managers, Medco, CVS Caremark, the pharmacy benefits managers that take care of your pharmacy benefits when you go to fill prescriptions are getting very active in this area of both having pharmacogenetic research questions as well as pharmacogenetic products. And these are just some headlines about how this is being done. And we have at the FDA a collaboration with Medco on these questions, and it's becoming very fruitful. There's a lot to be said by having a database of 60 million covered lives, 20 million of which have medical claims data. You can ask some pretty important pharmacogenetics questions in those data sets. So these are hot off the presses, and I think this is important to highlight. Physicians are ready for this. Clinicians are ready for this. This was a survey that Medco put out, brief questionnaire with the AMA, American Medical Association, to over 10,000 physicians. And they asked them questions about their comfort with pharmacogenetics, do they believe it could work, that sort of stuff. And these are the results. The overwhelming majority believe that genetics affects drug response. 2% didn't, which I'm interested to know who those 2% are. 23%, though, had prior education on pharmacogenetics. This is probably important to educators who are thinking about how to train medical students, pharmacy students, and so forth. 10% feel informed about pharmacogenetic testing. 12% ordered a pharmacogenetic test for patients in the last 6 months. 26 anticipate ordering it in the next 6 months. And 57 test not ordered due to not enough info. So the bottom line here is that people believe in it, but there may be an educational gap, and that needs to be addressed. Now, this is my response to change curve. This is where I see pharmacogenetics in any area, whether it's at FDA or in academia or in clinical practice. You have a few visionaries. These numbers are made up, but you have a few visionaries. You have what I have is about a quarter that are visionaries and change agents, really doing the work to push this thing forward. You have 65%, the majority, that are fence sitters. They're waiting for some proven benefit, or they want to be convinced. They can go one way or the other. And then you have functional resistors, 5%. These are people that are the devil's advocates, the ones that force you to articulate the value of what you're doing every day. We need these folks around. But then you have a small proportion that are non-functional resistors. They're not going to believe in it no matter what you do. And so what we're trying to do, and I'm sure what many of you are trying to do, is generate a critical mass here so that we can shift this curve right to the extent possible. And we know that this is happening. For example, this is the Cigna medical coverage policy for the warfarin or Coumadin Coumadin test. So, and there's some other pharmacogenetic tests that they're covering. So insurance companies are covering this stuff. Working to fill the science gaps. These are some of the FDA initiatives. So we have everything from our relationship with Medco to working in a guiding capacity really with biomarker type consortia, including the Biomarkers Consortium through Foundation for NIH, CAMD, which is mostly a neuro consortium. So this is Alzheimer's disease. Predictive Safety Testing Consortium, the International Serious Adverse Event Consortium, and some of our folks even have their own independent NIH funding to answer questions. So this is what I see as the critical path to personalized medicine. It's not really much different than the critical path to getting a drug approved. We need to learn about the disease biological pathway. Usually this comes in the form of preclinical or in silico or informatics type understanding. Development of biomarkers, understanding of structure-activity relationships, and in vitro testing is important. Then moving on to the preclinical space in the animals to look at some efficacy signals and toxicity. Using all of this information for rational dose and patient selection in clinical trials. And for those folks who were here for the earlier lecture, we showed some examples of how that's being done through modeling and simulation. Proof of concept studies and pivotal studies that build on this information. And then what I think is missing, and maybe it'll change with this new stimulus money, is healthcare practice and naturalistic setting research. So the medcos or the large clinical practices or even small clinical practices generating their own evidence that then feeds back into this loop so that the life cycle of personalized medicine doesn't stop when the drug comes to the market. So this is what the translational research paradigm would look like from my standpoint of clearly an understanding of candidates, whether they be candidate genetic markers or candidate pathways. And of course, there are exploratory approaches, genome-wide association studies, and some of the other things that your group is looking at now. And then, of course, you leverage all the information you have available, whether it's through in silico prediction, ex vivo data, in vitro data, some genetic epidemiology studies, drug-drug interaction studies, classic pharmacokinetic studies. And you really put that all together using sound understanding of research methodology, strengths and limitations of all these approaches, and synthesize these in a clinically meaningful way, in a practice-specific way, and in a patient-specific way. So with that, I will just throw these questions out. I have just 2 more slides. The questions for consideration. These are for you to think about, and maybe once you've figured it out, you can enlighten me, and I can pass the word on. How do we begin a realistic dialogue on levels of evidence? And you astutely raised this question earlier today. That is, what do we mean by clinical utility? What is useful enough to get these things in the hands of clinicians to be able to make decisions for people like my sister or people like you folks in the room? How do we get around this chicken-egg conundrum of limited clinical experience, limited recommendations? So it's not in clinical guidelines because it hasn't achieved a critical mass of clinical examples, but it hasn't done that because it's not in the clinical guidelines and doesn't give clinicians confidence to try it, so you sort of have this cyclical argument of generating data. What are the major challenges facing the clinical researcher focused on patient-oriented research? Or I would even extend that into informatics. And then this is, I think, is important. Should academic research fill the science gaps in a way that is legitimate to regulators, drug developers, diagnostic companies, and other innovators? Remember, we are looking to academic researchers to generate some of this evidence, but it has to be done in a way that's so methodologically robust that we feel comfortable making public health decisions based on that. So to the extent that researchers are always thinking about that, the public health implication, it doesn't matter what your research discipline is, if you're thinking about that, the likelihood that you can affect the change goes up. And so I'll end with this slide. I apologize for the redundancy for some of the folks that were here earlier, but I just really love this quote. This is Rudolf Buchheim, founding clinical pharmacologist, and he says, "A surgeon who uses the wrong side of the scalpel cuts his own fingers and not the patient. If the same applied to drugs, they would have been investigated very carefully a long time ago." So we would love to bring surgical precision to the way we use drugs. We know these things are not benign. They're associated with toxicities. And we know that there are dire consequences for not responding to medications. And so to the extent that we could use our armamentarium of scientific tools to get more precise about saying who the right patient is, what the right dose is, and what the right time in their disease process is to treat them, we would be doing a huge service to the public. So with that, I'll close, and I'm happy to answer any questions that you might have.
Questions?
Just one comment I have is that, like, maybe a great example of pharmacogenetics is the way HIV treatment is managed. Right, so like periodically, patients are their HIV their genotype for resistance mutations in HIV, and then their medications are modified accordingly. So
it's a very astute observation, and in fact, think about the way we treat HIV patients on a global scale. So not only do we use viral genetics, but we use viral load, right? So clinical decisions are made on biomarkers. And the story behind viral load is the same story behind genetics. There's nothing special about genetics. So we should be putting our hats on and thinking about the way we've dealt with biomarkers in the past. The HIV one is a perfect example because you have something like a Bacavir, right, which is where you're testing the patient's genome to say something about the likelihood for risk. But you have drugs like Maraviroc, which is a CCR5 receptor blocker, where you actually are looking for viral tropism and whether or not the drug is going to work based on viral properties. So it's actually a perfect example. You're using biomarkers, you're using the host genome, you're using the viral genome. There's some examples for HCV as well. There's for hepatitis C, common therapy is PEG interferon and ribavirin. And depending on what your hepatitis C viral genotype is, you get, I forget what the exact numbers are, but let's say it's something like 3 months of therapy versus 6 months of therapy. I don't know the exact numbers, but duration of therapy depends on your viral genotype. And now a series of genome-wide association papers show that the IL-28 beta variants actually correlate in the patients, not in the virus, correlate with response to those therapies. So another example where a clever clinician not only takes the viral information, which is in the drug package insert, but then also takes the patient's information and says, based on these characteristics, I think we should be aggressive, or we shouldn't try these therapies because they're associated with things like depression, high blood pressure, and those kinds of things. So I actually think the viral world is where a lot of this stuff might get applied in the future. And it has a lot of those factors that we talked about, patient activists, clinicians who are savvy and deal with life-threatening diseases, clinical guidelines that have the information in it. So I am enthusiastic about that.
It would be nice to see that culture propagated to other diseases and the problems.
Any ideas how to do that? No, you're right. I mean, I think if anything, there's a subtext here, and that's culture, right? So I showed you the data from the cardiology world. Cardiologists on the whole, think they're doing it right, but there may be a culture behind that that might not be the same culture for infectious diseases.
So I found the slide with the refuted interventions really interesting, actually seeing how long it would take for some things that may not be useful interventions to be phased out. Any thought on what the balance in personalized medicine system is going to be between introducing new therapies to targeted populations and saying that this specific intervention makes no sense for specific subpopulations?
Yeah, excellent question. So, I can probably spend an hour talking about that question, but let me just give some thoughts off the top of my head. One is we have to be very careful in pharmacogenetics about what you're saying, because I think there's going to be a lot of publication bias in the world of pharmacogenetics. So things that we found to be things that have been repeatedly reported, we don't know how many neutral studies are out there that would have refuted them. Now, granted, we have some quality controls now. You can't get a paper published these days unless you have an independent validation cohort that shows the same thing, or at least a lean in that direction. So we're trying, we meaning the academic community, are trying to prevent those false positives, if you will, from driving our decision-making ultimately. The second question about targeted therapy, or the second aspect of your question, is one that we're really thinking about now. So you have a drug that's approved only in marker-positive patients. Okay? So how much data would you like in the marker-negative patients to know if there's maybe some responders there, right? So this is something that we are thinking about. It's something that we're trying to develop guidance around, at least from the drug development standpoint. But I think if there's a demonstrated benefit in a subgroup that was pre-specified, in other words, if let's say you only studied marker positive, but your drug worked compared to placebo, it is what it is. The drug worked. You can use it in that population. The question of added benefit in other populations is, I think, a very important one.
I guess also removing risk. I'm thinking of the recent breast cancer mammography screening guidelines, where you have an intervention that is saying that this is a good intervention, but it makes sense to restrict the group that you're applying it to. So it seems like there are huge non-scientific factors that will enter it also.
Let me say that I haven't read those guidelines, but I'm familiar with the issues. And what they illustrate to me is a couple of things. One is, first of all, I think there's an inaccuracy in reporting of information. So I think those guidelines didn't say that testing is inappropriate. I think it said routine testing is inappropriate. So people leave off the routine part, and it's not a mandate to say don't test in this age group. So I think that was a headline-getting thing. But the other thing that we need to be sensitive about is we are pretty arrogant about what we think we know about the genome. I mean, you know this. At one point, we thought 99.9% of the genome was junk, right? Because we couldn't figure out what it was used for. So we have to be, I think, humble enough in this emerging science to revisit the evidence as it emerges. And I think that's what happened in this mammography screening thing. I don't know the specific science behind it, but it's an illustrative tale of how we need to be okay with revising our recommendations as new data emerge.
A question on other therapies. So pills is one thing. The emergence of realizing that many diseases have a regulatory effect, the subtle and is not going to be done with chemicals, maybe controlling expression, maybe injecting DNA, maybe, what is the state of the, what did you say you were screening the horizon?
Yeah, horizon scanning.
Horizon scanning
right.
For those kind of things. Are they coming?
Well, so typically you're absolutely right. The market has been small molecules. I will say, though, that biologics, drugs that are therapeutics that are developed through biological engineering, cells, is representing a huge growth. Something like 20% growth in biologics versus 7% for drugs. So I think biologics is a big area. Some of the other innovations, like gene knock, siRNA type therapies, gene therapy, they have problems. Especially gene therapy, siRNAs, people die. So I would say that we're, from an agency standpoint, that we are supportive of that stuff. And of course, we will look at it when it comes in, but those aren't coming in. People are not bringing in gene therapies for approval. So I think there still needs to be some work done on some of the technologies like gene therapy. But clearly, siRNA and biologics and other treatment modalities.
How about DNA? Kind of intervention based on actually DNA or things that are by definition non-toxic because they are pieces of the or is toxicity. I heard those kind of things that, for example, cancer overexpression could be controlled by injecting DNA regulatory pieces such that the binding of transcription factor will be competitive to the extra DNA and slow down. So it's a very interesting dimension of treatment and therapy as opposed to just molecules.
Absolutely, and to the extent that that's in the early phases, we don't think too much about it from a drug approvability standpoint. But what I can say is that there has been a growing, I didn't show this slide, but there's a lot of growth in terms of the types of genetic applications that we're seeing within FDA, the way drug developers are applying, let's say, genetic information in their development programs. So with that recognition, there's been a desire to build infrastructure, regulatory review infrastructure, to deal with these emerging sciences, whether it be an informatics infrastructure, biomarker databases, getting clinical reviewers and other disciplines just familiar with the jargon of the field of genetics. Those initiatives are ongoing. So if these therapeutics prove their concepts in humans, I have no doubt that they would be coming forward for regulatory review.
At this point, they are not knocking on the door in large numbers.
I don't even think they're in the driveway. Right. Not that I'm aware of. Other questions?
So since you're relying a lot on the body of academic literature to kind of draw inferences about known pathways and mechanisms, it does seem like publication bias could potentially be a huge confounding factor in the way we think about drugs. And it seems to me like one big problem is that studies don't get published unless you have a positive result. Right. So, you have 100 studies, like 99 show a negative result, the one that shows a positive result gets published. So wouldn't maybe one thing we could do to help this is to have some journals of negative results?
There actually is a journal. There's a journal called, I think it's something like the Journal of Non-Significant Findings or something. It's one of those open access journals. Oh, yeah. No, you're raising a valid point. So there are a couple of, let me just clarify a couple of things. I think that the information that's in the public domain is relied on in two ways. One, drug companies that are trying to figure out novel targets for therapeutic intervention, for the most part, are not doing genome-wide association studies because, as you saw, there's so many being published at any given time, and the data are publicly available, that they use their own informatics work or work with statistical genetics people to identify what might be these druggable targets. So I think from a drug, early drug development or discovery standpoint, I should say, I think drug companies rely on that info. On the back end, we rely on that info for already approved drugs to see if we need to make some revised statement about risk and benefit in a subpopulation. We don't take that lightly, so we won't take, if it's a single publication out there, it's not going to even probably make it on the radar screen. As I mentioned a bit earlier, now there's a convention where you can't even get something published unless you have replication in an independent cohort. And if there's a bunch of those papers with a bunch of replications, then we start to get more serious about it, unless it's something like the safety thing where we don't reasonably think we're going to get much more data than what's out there. In drug development, if a sponsor wants to say something in their drug label for marketing purposes, we're very rigorous about that. We make them generate the data, and we analyze the data. So there's kind of a spectrum of how you might use this information throughout let's say, the life cycle of a drug or a biologic. This neutral finding idea is a good one. And there are ways to assess for publication bias through meta-analytical techniques, and we try to look at some of those as well. But you're making a provocative point, and I really do think there's a journal of, I think it's called Negative Findings or Neutral Studies or something like that.
Well, I think physicists can publish any experiment that will not work because they say we learned not to do it this way. But mathematicians, have 1,000 false attempts to prove something that you can never say, I tried this. You see that in this area, this bias that Austin is saying is extremely valid.
Absolutely.
Not only that, but with the pressure of grants and to have a positive result, you may cut corners just to get a positive result. So, you try 10 times something, and 1 in 10 is not very impressive. So he said, I found this, but I found it 3 times. And all of a sudden, it's a huge, beautifying result. There is no way to check what the investigators.
Well, especially in pharmacogenetics, I think that the trend is to do high throughput, medium to high throughput genotyping, and then look for interesting things, right? Because unlike with diseases, pharmacogenetics is tough to do because how do you find clean populations of drug-treated patients and that sort of thing? I would say that the added danger to what you're saying is not just the false positives, but not knowing what's out there, what's been done and tried and didn't work, leads to a lot of redundancy in research questions. So if you're paying $500 per genome-wide scan times 1,000 patients, that's already $500,000 for a question that maybe didn't need a genome-wide scan because we had some prior knowledge that never made it to the public domain. So I think that resources are becoming a critical factor, and to the extent that we can get as much out there, even if it's neutral, that would be relevant. Well, I thank you for your